Yeah.
Megan?
Yes.
Was Carol part of the invite?
Yeah, she got it.
All right, well, let's go ahead and get started. Thank you, everybody, for joining us once again for the next edition of Physiology for Administrators. Today we get to hear from Dr. Achim Klug, who is a fascinating individual to work with and get to know, and I think you'll really like his. I think you'll really like to hear what he has to present us here shortly. So Achim graduated from the University of Würzburg. Am I saying that right?
Yeah.
Oh, I did. In Germany, where he got his bachelor's and his master's. At least that's the American equivalent to what he got there. He got his PhD from the University of Texas at Austin. Hey, Chris. And he did a postdoctorate at Oregon Health Science University in Portland, Oregon. Achim joined our faculty in 2009, and so he's been with us quite a while now, and was promoted to associate professor in 2015. And he is one of our hearing researchers, so you'll get to hear, no pun intended, what he talks about, or what he's researching. So Achim, I'll go ahead and turn it over to you.
Okay. So we will talk about how fast neural inhibition in the auditory system helps you at a cocktail party. And why do I say cocktail party? We'll get into that in a moment. I want to start out with a very fundamental and basic question that I and a lot of my colleagues really wonder. How does the sensory system process information? How is it that you look at me and information, visual information enters your eyes, and these are all pixels, and then you can make out people, cars, things coming at you, running away from you. When you think of the auditory system, there's just random sound waves that come into your ears, not just mine, but also the AC and whoever happens to make a noise. And how is it that your brain can sort through that vast amount of information, that stream that comes in all day long, and extract what you're interested in? So that's, of course, a really big, complicated question. And after having done this for decades, I still don't know how to answer that question, because we understand so little about the brain. But the first point is that the auditory system is a pretty good system to study this question. And the reason is we have these anatomically separate sub-areas. So these are some auditory nuclei, the first 20 or so. In the ascending system, here is the ear, the cochlea, this is the auditory nerve. And then information goes from one area to the next, and it ascends generally from here to here. And so these are very spatially separate areas. There are a bunch of neurons in this area that they all do the same job. And then there are a bunch of neurons over here that do a different job, but all neurons within this area do the same job. And so we can break down that complicated question, how does the auditory system do it, into 20 sub-questions and say, how does this guy do it? How does this guy do it? And that guy over here. And these are much simpler questions that we can actually address. And then what we want to do is basically we want to look at an input to these areas. Action potentials are the fundamental piece of information in the nervous system. I'm sure my colleagues have mentioned that before. So it's kind of like ones and zeros in the computer. This could be a 1. An action potential, this is a 0 here. And you have streams of these ones and zeros crisscrossing around the brain. And each neuron receives inputs. They look like this. And the neurons apply certain sets of rules to that and change the input into a different output. So the whole point of information processing is that the output is different from the input. If the output were always the same as the input, we would be really stupid and life would be really boring. We would never get anywhere. And so the whole point of extracting information is that the output is not the same as the input. Computers do that also. They have transistors that are called AND gates and OR gates. And the whole point of that is that you have information and you change it, and the output is a different one. And then the next nucleus gets this output as an input, and they use that and add another layer of information on top of that. And so you go your way up from the ear to the brain in maybe several hundred stations, and you can imagine how things get really complicated very quickly. But at the same time, this is an enormous tool to process information. And when I say set of rules, what we mean in neuroscience is we want specific information about the neurons, the wiring, and the types of connections. So if we understand these 3 things, then we understand the rules that underlie the conversion of this train to that train. And so that's basically what I study as a biologist. And the area where we do it is, next one, is this area down here, the sound localization pathway. So we're very close to the auditory nerve. We're only about 2 synapses away from the nerve. And the first question is, why do we even need a sound localization pathway? What's the big deal here? And so the reason is that there is no spatial information in sound that comes in. So if you think of the eye, this is my rendition of the eye, and the retina in the back is a projection screen of space. So if I look at you, I have an upside down mirror image of this room in my eyes. So without trying too hard, spatial information is right there. Whatever is bottom left will end up top right on my retina. So information is spatially sorted. Sound doesn't do that. Touch does that. If you close your eyes and I poke you at your arm or your leg or somewhere else, you automatically know where I poked you because there are wires going from various places in the skin to the brain. And every place in the skin is connected to a different place in the brain. And so space is inherently coded with that information. Sound is not doing this. Our ears do not have any spatial information. If you had only one ear, you could never tell where a sound is coming from. But fortunately, we have 2 ears for that purpose, and a lot of other animals too. And what happens is, whenever there is a sound, you receive sound information with 2 ears, of course. And when the sound is right in front of you, the sound arrives at both ears at the exact same time because this path is the same distance, and it will also arrive equally loud between the 2 ears. And when the same sound moves over to the left, then this path is a little shorter than the path to the other ear. And so sound will arrive a moment earlier than here, than over here. And to appreciate this, you also need to know that sound travels pretty slow through space. Like the speed of light is about halfway to the moon in 1 second. Sound travels only 350 meters per second from here to Colfax, which is Peoria. So it's much, much slower than light. And so we can actually calculate, now that we know sound travels 350 meters per second, we can calculate how long would it take the sound to travel from here to here. For a human head, 20 centimeters, that is about 500 microseconds or so. It's a small amount of time, but it is not unmanageable. With light, you could not do that. And because we have information between these 2 ears, and because there are these time and intensity differences, so time difference because that path is shorter, and intensity difference because the head is simply just another object in the way of the sound, and it obstructs some of the sound waves that come from here. So if you put a microphone in this ear and another one in this, you will find that the sound is less loud over here. The ears, of course, are microphones. So they sense time and intensity differences, and if you can extract these, then you're in business for recreating space. So the auditory system creates space with computational efforts by extracting time and intensity differences. And that's done in the sound localization pathway here. And so these are 2 main nuclei that extract time difference, intensity difference. That's the one we study. It provides inhibition to both of these, and not surprisingly, neurons here and here have to be connected to both ears in order to be able to make that comparison. And so the biologist in me is interested in that question because, bats localize sound, owls localize sound. It's a very interesting biological problem. How does this work? There is also a technological and a human health component to that. So the technological component is, we're going to watch 2 movies that are very brief. The first one is a commercial from a company named Audience. And so those of you who have had cell phones 10 years ago or so, maybe you remember those dumb phones, as they called them back then. It was horrible to have a conversation in a loud room like a bar or at a busy street. I remember, kind of trying to have a conversation with my landlord when I just moved to Munich at a busy road, and I was kind of in some house entrance and holding this ear, putting a finger in this ear. And we don't have that problem anymore today. And the reason is because of this chip here that's made by a company named Audience that is in all the Android phones. Apple, the iPhones have a very similar chip. They don't use that company, but they use the same thing. And what they do is they basically exploit the same principles as the sound localization pathway and put them in a chip. And it filters spatial channels. It filters the sound that is around here according to spatial channels and focuses on the one you're interested in. And that really helps you to have a conversation. So here is 40 seconds from their commercial, and I could not say it any more blunt.
About the freedom to stay connected wherever you go. But in the noisy world around us, sometimes having a conversation can be a real challenge. Now there's a breakthrough technology that delivers clear mobile calls, better voice quality, wherever you want to talk. Introducing EarSmart, the world's most intelligent voice Equipped with this tiny chip, your phone can deliver clear conversations in all situations. So you have the freedom to make and take calls with unsurpassed call quality just about anywhere. This revolutionary
So what I like is how the phone becomes the ear and the head with two microphones. So basically what they're doing is exactly the same principle. They're using two microphones, which are the two ears, and they use that to make spatial channels from the information that comes in, and then the phone knows which spatial channels are the important ones, like the voice, not the noise, which is in a different location, and it filters through that and helps you make clearer calls. So that's not directly from our research, but this company was based out of Wisconsin. There's a big auditory research group where their tone comes from. And so somebody over there put two and two together some years ago and said, hey, you know, we should apply what we know about sound localization to technology, and then they founded this company. So then where we come in more is in the next step. So when I talk to you now, there is not only the first wavefront, as we call it, like from my mouth to your ears, but because we're in a room that has walls, ceilings, tables, hard objects, the sound also goes in all sorts of other directions. It bounces off this wall, it bounces off the table, so you get multiple wavefronts. And you should be really confused about where I am standing, but you're not. Close your eyes, you can point totally exactly to where I'm standing. And the reason is your brain, the sound localization pathway, also can deal with all the echoes that are coming. And so that was also a huge problem for engineers initially. And so that's something that we did in Munich. We figured out how this echo suppression circuit works and how you can localize in an echoic environment. So a student, this guy here, these two people, one was a physiologist, one was a computer scientist, and then Thomas, he took a teddy bear, literally a teddy bear, and he took out all the things from the head inside the teddy bear, and he put two microphones in there, and then he asked the teddy bear to localize sound. And he had a computer sitting there, and when he did just the plain sound localization, it was exactly what I told you, like it was the first wavefront. A moment later, all the others, and teddy bear was really confused. I mean, he was confused in enclosed rooms. Like if you put him outside in the grass, teddy bear did just fine because there are much fewer echoes. And then he went to the bathroom because it's tiled floor to ceiling. That's the room with the most echoes. And teddy bear was really confused, and it flipped the switch on the computer and turned on that circuit that he described. And all of a sudden, teddy bear could localize. And so basically, here's another thing that was copied from nature. How do we localize in an echoic environment? So here's the biology paper, and then about a year later or so, Honda Corporation had this paper which said modeling the precedence effect. So Honda has this computer, this robot named Asimov. You've heard of that one? So it's arguably one of the most advanced pieces of technology. You can watch Asimov for a little bit here. So it's a household robot. So yeah, so Achim can catch balls, can jump, can walk stairs, can serve you tea, can pour water in a paper cup, catch balls, kick balls,
and dance.
And dance, yeah.
This was key in his programming.
This was key. So if we go a little bit further,
Cool.
so,
Rob, we might need one of
paper
those.
cups and serve them. So each one of these tasks is incredibly complicated. If you ask engineers, if you fill water in a paper cup, how do you not crush it? And if you don't hold it tight enough, it will fall down. So that's a very, very difficult problem. Walking stairs, jumping on one leg, which we do all the time. Very hard problem for a computer. And so where Honda wants to go with this guy is basically the household robot that could make coffee for you, serve you tea, get the newspaper from outside. And you can't buy these. They're a little expensive at this point. They cost about $1 million, I think. But maybe prices will come down like with so many other things. And then Honda had the same problem. Their robot could not localize. So when you're standing in your room, you say, hey, Asimov, and then Asimov doesn't know where the sound's coming from. But they wanted the robot to turn and look at you to wait for its command and just function in rooms, because that's probably where it would be used most of the time. And they had that same problem that Asimov could localize in closed rooms. And they said, hey, here's something, this is how nature does it. It was just published. And so the Honda's paper basically says, we copied that exactly how it works. So here's the technological aspect, but there is also a human health aspect. And that is, you use your sound localization pathway typically not to hunt or be hunted, because that doesn't happen that often in the modern world, but you use it whenever situation like this. You go to the cafeteria at noon, the bar on a Friday night, situations like this where many people talk. You use your sound localization pathway to make narrow spatial channels and then focus on the conversation you want to have. Think of this person, she wants to talk with her friend here and not talk and not hear or filter out what all these other people are saying. And as it happens, there are a number of medical conditions where these spatial angles get wider and wider because the circuit doesn't work so well anymore. And then people are really confused. And so we study these, and one is central hearing loss, which is a type of age-related hearing loss. And another type is autism. That's the most of the business contribution to the lab at this point. So basically, if you talk to somebody over 50 or at retirement age, then the chances are 50% that these people would have immense trouble functioning at an environment like this. And 10 years ago, that was completely not understood. Everybody knows that people need hearing aids as they get older. Some people do. And that is because the cells in your ear that translate the sound into action potentials, they die, they do not regenerate. And so the day you had your best hearing was the day you were born, then things were going downhill from there. And every time another cell dies, you hear a little less well, if you will. And at some point, maybe your hearing thresholds get so bad that you need a hearing aid or a cochlear implant or all these things. But all these manipulations treat the level of the ear. There are a lot of people out there who walk around who have perfectly normal hearing at the level of the ear, but they still can't function in these environments. And 10 years ago, they would go to the otolaryngologist and say, yeah, doctor, I was at this wedding on Saturday. I could not talk to my relative. Something must be wrong with my hearing. And they would give you a normal hearing test where they put you in a closed room, sound isolated room, and then they play sounds of various frequencies and intensities. Every time you hear something, you push a button, and the result might be that your hearing is perfectly normal. And then they were at a complete loss what to do. And then some people were sent to the psychiatrist to be checked out if they're crazy. These things really did happen. And today we understand there is a completely independent change, an independent mechanism of age-related hearing loss that is in the brain, in the sound localization pathway. And as the circuits change with age, the consequences that these spatial channels that you can isolate get larger and larger. And if you have a person who is suffering from central hearing loss, then if she were suffering, she may not be able to separate between this voice and that voice and that voice. So if these people talk at the same time, then they can't filter through the three different voices effectively, and they're confused. They can't have a conversation. And so because this condition affects so many people, up to 50% of the population at retirement age, it's a huge health problem. You won't die from it. But it's very frustrating for the people. There's also a lot of evidence that things like dementia go along with this. So because people avoid communication and because people can't keep their brain on its toes, if you will, by constantly having useful, important information coming in, if you can't do that very well, then secondary medical conditions, depression, dementia, Alzheimer, are following with a much higher probability. So it's one of the central problems of healthy aging, as people call it these days, and we need to fix it somehow. And so I would then say the work in our lab is basically three areas right now. So the basic function, how does the sound localization work? That's the biologist, and we work with techniques called patch clamp, video recording, behavior, and anatomy. We'll get into all these in a moment. And then we also want to know what are the age-related changes in that circuit. And so that's partially in collaboration with the clinic, the human studies, there are also patch clamp and ADR recordings, it's also collaboration with them. And then we look into methods of manipulation. Optogenetics is maybe something that some of my colleagues have mentioned before. Virus, you hit the brain with the virus and you try to express new proteins, glass virus, engineered devices, and these are seemingly unrelated projects. If you talk to somebody in the lab, then they work either here or here. But in my mind, they all converge on one point, maybe 10 years or so in the future, which is at the treatment of central hearing loss. So we're finding out how the circuit works. We're trying to study how it changes, and we're studying methods to manipulate it. And then all these, when you put these together, then maybe we can find a way to treat central hearing loss. And we have some ideas where this might be going, but it's still some years out. Yeah.
Oh, the picture behind, is that something related to the ear?
Actually, this is the brain area we study.
Oh,
We just
okay.
used it as decoration so far. These are the cells. These are neurons in the sound localization pathway, and they receive excitatory and inhibitory inputs, the red and the blue ones.
Ah,
So these are synapses that come in and bring information into the cell. And so, and that's called immunohistochemistry, the technique with which this picture is taken. So your immune system is really good at attacking particular proteins. So before you were even born, there were like billions and billions of different antibodies made randomly in your body. And the idea randomly is because we can't catch up with viruses. We need to have a whole set of treatments or keys available to viruses that are even being evolved 10 years from now. And so what you could do is, if you can isolate a particular antibody, an ion channel or something, you can isolate that protein, we could inject it in you. In practice, we don't do humans for this, but we use sheep, goats, mice, or other animals, and it literally gets injected into the bloodstream of these animals, and chances are high that there is one antibody in this animal's immune system that fits perfectly for that foreign protein. And then it creates an immune response similar as if you have a cold, where it takes about 3 days or so, 5 days for a cold to get over it, so that there's one antibody that fits that cold virus perfectly. It happens to be there because it was built before you were born. And then because it found its target, it gets amplified over and over again. That's the immune response. And then it fights the virus, and that's why you get healthy again. So if we inject some foreign protein, and in this case, these were synaptic proteins of excitatory and inhibitory synapses. And we didn't do this, we buy these, but this is how it's done. So you inject it in an animal, you wait about 10 days or so, and then you draw blood from the animal, and then you isolate that antibody, and then companies sell it and you buy it. And at this point, there are thousands of antibodies out there against all sorts of proteins. Some labs make their own. Rock, for example, does sodium channel antibodies. It does exactly the technique that I just told you. And then you tack fluorescent proteins on it, and then you can buy several antibodies against several types of proteins. You get these multicolored images. And what it allows us to do is to quantify exactly how many excitatory inputs are there, how many inhibitory inputs are there. You can count them, you can measure them, and you use the confocal microscopes for that. So they have laser lines of various colors in them, and like this one is excited by a particular light frequency, and then it emits in red, and you catch that with a camera, and then you switch to the other laser, and you excite the blue ones, and then the green ones, and so forth. Cool. So that's what the complicated microscopes are doing over there. And you had a question.
Yeah. You mentioned the relationship between central hearing loss and dementia or depression and other neurological diseases. Is that something that's currently being studied beyond like what you're doing, and is there been any kind of cause-effect relationship figured out yet between central hearing loss?
So there is a lot of work, if you look in the literature, as what is the probability of getting this disease, and that there is cause and effect as the exact mechanism we don't know yet. But we're at the point where we think it's not a coincidence that somebody is just more sick than somebody else and they have hearing loss and independently dementia, but one results from the other one. And depression is an obvious one. People who don't hear well are often depressed. Because they can't communicate. Like if you ask anybody in the street, would you rather be deaf or blind? Most people would tell you, blind would be a disaster. I wouldn't be able to find the door over here. So I would rather be deaf. But fact is that people who are deaf are much more isolated than people who are blind. If I were blind, I could be having this conversation with you the same way. If I were deaf, it would be a lot harder. And so depression is an obvious one. People feel depressed because they feel isolated. And then the dementia is what people believe is the brain is not challenged enough. If less information comes in, auditory information, the brain is less challenged. And that's maybe if you stop reading books and watched stupid commercials all day long for the rest of your life, your IQ would go downhill, and dementia and things would set in. Like basically, if you don't challenge your brain, same as with your muscles, if you don't exercise, then your muscles decay. And brain is a similar case if you don't challenge it. So that's how you might explain the dementia. In Alzheimer's, there is a big link there, but the cause, we don't know that.
That slide that you had the 3 different
area.
This one, yeah.
Where does Liz's autism research
So-
fall? I don't.
You know, so it would fall in this one because it's very similar to the age-related changes, but it's also, I'm kind of leaving that up to her mostly. So it's kind of a classic example of a postdoc being in training and then they have to find their own niche, which they take with them when they leave. And so that's it really. So we're only pursuing it for now, but the way I see it, that she would be mostly the driver of that when she leaves the lab, but it would fall kind of this one here. Okay, so how does this? So how does sound localization work? I told you earlier that we're isolating. The brain needs to isolate time differences between the two ears, and so how would the brain do that? So you have a right ear and you have a left ear. And so imagine the sound comes from the midline, so it arrives at the same time at either ear. And then the action potentials start traveling down this nerve fiber, this axon, at the same time as they start traveling down on the other side here. And so the first important component of this model here, which is called the Jeffers model, is delay lines. So you kind of find an internal representation for the external delay of the sound. Like if the sound comes from here, there is a delay of its arrival to this ear, and you find a fiber in the brain, or you make one, where action potentials travel with exactly the speed it takes for sound to go around the ear and hit the other ear. And the next important component are these yellow things. These are cells that only send an output if they receive input from two sides at the same time. So it's an AND gate, as the engineers in computer science call it. So this cell will only have an output from here and here at the exact same time. And so with these two things, you can localize sound. So imagine a sound from the midline, it arrives here and here at the same time. So the action potential travels along here and along here at the same time. It will go over to this route and will hit cell number 5 pretty early on, but nothing will happen because the sound is only, because the action potential is only this far down the other side. So there is no coincidence. So left ear action potential will arrive here at the same time as right ear will arrive here. Nothing happens. Then the next impulse will arrive here and here at the same time. Nothing happens again. And then cell number 3 is the one where coincidence happens because that path here is the same length as this one. So cell number 3 represents the midline. Now, if a sound comes from the left, from the right side, it will arrive at the right ear earlier than at this ear. The action potential will start traveling along here before it even hits the other ear. And so the coincidence is maybe at cell number 1. And so cell number 1 represents the left side, the right side, and cell number 5, the left side. So if you look at this array of neurons, that's a map of space, basically. Right is here, mid is here, left is here. So if you ask the little man in the brain, where does the sound come from? The man will look at which cell is active here, and then say, very far right, midline, a little bit to the left, or something like this. So that's how sound is being localized. And the time differences are very, very small. So 500 microseconds is the largest time difference you would ever experience as a human. That's for sounds coming exactly from here or from here. And you can not only do left from right, you can also do a few degrees in space. So you can probably do 10, 20 microseconds time differences. And considering that an action potential is already 1 millisecond long, we're 2 orders of magnitude faster than the unit of information in the brain. That's a very interesting problem, that the brain has to code for some stimulus, some space, some event in space that is 2 orders of magnitude faster than the unit of information. So not surprisingly, everything is extremely tuned towards temporal precision in that circuit. Big fibers, big synapses, everything is very fast, very temporally precise. And when that temporal precision declines with age, that's when the central hearing loss happens. And so that's basically the assumption right now, that those connections in the brain change a little bit, they get a little bit slower, and as a result, 2 degrees in space become 20 degrees in space, and then you have central hearing loss. And so how do we study that? Yes?
So based on a model, how would a blind person determine if the noise is coming from in front or behind.
Oh, okay. So that's a very good question. So this is only with one single frequency you can't. But natural sounds have several frequencies usually. And so if you only have one frequency, then the sound, you say the sound comes from over here, but really it could be at any point along a circle like this. It could be from behind, the front, it could even come from top. But if you have two frequencies, you get one circle that looks like this, and you get another one that crosses that first circle at 2 points. Then you narrowed it down to 2 points, and then if you have a third frequency that also has its own circle, and that intersects with the other two only at exactly 1 point. So you're absolutely right. For single pure frequencies, people get confused. They cannot tell the difference front to back. But a pure tone kind of only exists in the lab. It's like you have to make it artificially. Every sound, even a pure tone from an instrument, already has multiple frequencies. It has harmonics in it. So a pure tone really only exists in the lab. So in natural life, you can get around that problem just by having multiple frequencies.
So Achim, is that
Yeah.
happening in the brain, or is that
That's in the
happening-
brain, yeah. So
Oh.
that's in the brain, and it is happening in that guy over here.
Oh, that's the brain. I
So
thought that
exactly.
was part of the ear.
So no, the ear is here, the
Okay.
cornea, the auditory nerve, this is the brain. So this would be a picture if you cut off somebody's face and look into the brain.
Okay.
And so that area here is which has the
Comer doesn't approve of that. by the way.
Yeah.
So, yeah.
purely hypothetical. And so yeah, here's one input, here's the other input. And then what we study in the lab is, so the Jeffers model only has two excitatory inputs. The word inhibition, I didn't mention it because the Jeffers didn't mention it, but we know from anatomy that from the picture we discussed earlier, the chemistry, there are inhibitory inputs to these cells. And so what are they doing? That's what we study. That's part of like the biology area. We use patch band recordings. For that, patch band is a technique where we can take a brain out of an animal and slice it into sections very quickly. The sections stay alive for about 8 hours or so after the head of the animal comes off. So this is a section of live neurons, and this is how they look like. And then you can stick glass electrodes into them and record electrical currents, such as action potentials, but also finer detailed currents and synaptic currents. You could even isolate single ion channels with that technique. So that's patch clamp. Another technique we use is in vivo recordings. So the advantage of patch clamp is that we can look at the cells, we can see them, we can analyze them in great detail. The advantage is the ears and the auditory nerve is cut off. So if you study hearing, that's a disadvantage. So what we would need to do for that is to give fake trains of action potentials and hope that it somewhat resembles what the brain is doing. If we record in vivo means we work with a whole animal. You basically drill a head in the skull of the animal, and then you advance something like this into it, and you hope to point that tip of the electrode very close to a neuron. Then you can record action potentials also. So you work with a whole animal. You can actually play sound to the animal. That's a big advantage. But the disadvantage is you cannot analyze the cells in very great detail. And so all techniques have their pros and cons. And by using several side by side, you get, in my opinion, the strongest results. If you can show the same thing with multiple techniques that converge on the same point, that's relatively strong evidence that you're on the right track. And then we also use neuronal modeling, not nearly as high leveled as Joel does. So we use modeling as a reality check. So if I can make a computer program that can simulate information processing in a particular brain area, then I can ask this computer model, I can give trains of activity into that, put into that, and ask the program to tell me what the output should be of that area. And if that is correct, then I know I'm on the right track. So it's a kind of a reality check. So here are some synaptic amplitudes. And one of those lines is the actual data, and one is what the computer model said. So it's not perfect, but it's 94% perfect. So it's pretty good, and that tells us we're on the right track. So we're not doing very sophisticated, high-level models as Joel does, but we use it as kind of as a reality check.
So Achim,
Yeah.
if your ears are really just a hole for your brain to hear, then your ears aren't really for hearing.
Well, the ears are for hearing. So I skipped that part because we don't study it. But the ears, there is a membrane in here, the tympanic membrane or eardrum, and that vibrates with the sound. And then there are little bones called ossicles. They get vibrated also. This is the middle ear. And then they push into a fluid-filled space called the cochlea. So the little bones is like a little hammer that goes into a stretchy window of that fluid-filled space and pushes into that fluid-filled space with the same frequency as the sound vibration. And then there are cells in there that translate the vibration into neural activity. And so they, but I skipped that whole part.
Dan studies that.
So the cell,
Yeah.
Dan should talk about this. That's more
Dan
like his area. So the cells look like this, and they sit in a membrane here and another membrane here. And then as it vibrates, these two membranes shear against each other with the beat of the sound, if you will. And then there are ion channels here, little holes, and then little chains. And so whenever the stereocilia are bent in one direction, so I'm not doing any trick, I'm aligning these pins such that one lid is next to the white area of the other one. If you move it this way, you see how the distance gets larger. I didn't play any tricks, that's what happens. If you move it the other way, the distance gets shorter between them. So if you had a little chain that connected these two and a trapdoor, like imagine your attic trapdoor where you pull the chain, it opens, you let go, it closes again. So that would be in here, and then as this thing vibrates, it opens and closes that channel, ions come in, and that's how you get from vibration to the action potential. So that's happening in the ear.
And then does it also do balance?
Yeah, they're the same type of channels in the vestibular system. That's not in, I guess technically it's part of the ear also, like the semicircular canal. We don't study that at all, but the same type of cells are there, and as you move around, there's water in them that sloshes back and forth and moves those stereocilia and opens channels, and that's how you sense motion and balance. Okay, and so I would like to spend a little bit more time with the age-related changes. So what we can do here is record a human's ability to hear, if you will. So this is an ADR trace auditory brainstem response. This is done by putting electrodes near the back of the head about here onto a human patient. You take duct tape or band-aids and you tape them on, and then you play loud brief sounds. And the principle is of a loud brief sound is it excites all the cells in your ear at the same time, like all the hair cells do this at the same time. And then there are action potentials in the auditory nerve at the same time. So if one cell fired an action potential, you could never measure it with a piece of wire that is taped to the outside of the skull. It's way too small, but if many, many cells fire at the exact same time, you can measure it, and then you get something like this. This is wave one. So that's the activity in the auditory nerve, measured from a wire taped to the back of the head. And then a moment later, this guy here fires these cells. That's wave two. And a moment later, wave three fires, which are these guys here together. So we have a non-invasive, easy method to measure activity in these various brain areas. Then number 4 is this guy, number 5 is somebody even higher. And so non-invasively, repeatedly, as often as you want. And so that's called the auditory brainstem response. You can also do it in animals. We get to that in a moment. And so what we did in the clinic is take old people and young people and do ADR measurements. So here's wave 3, the one we're interested in. I want to draw your attention to wave 3. See how nice and sharp, tall, and thin it is here, and how wide and much narrower it is here. What this means is that cells in these areas here, fire very well synchronized in young people. That's a 21-year-old person, that's a 68-year-old person. And so when all the cells fire at the same time, all the action potentials line up like a tower, and you get something like this. And when they're all temporally a mess, then you get something like that. So what this tells us that specifically wave 3, and that's the one that was significant, is lower in older people, but not in any older people, specifically in those who report hearing and noise problems. So we gave everybody a questionnaire, all of our volunteers, and asked questions like, does hearing problem make it difficult to carry conversation in places where many people talk at the same time? And then we looked at the ABRs of all those people, independently of age, just who said they have problems, who said they don't. Not surprisingly, the 21-year-olds never had problems, because that's typical. And the 68-year-olds or the 65-plus-year-olds, 50% had problems. So rather than going by age, we sort them by how they answered that question. And it was even more significant that this wave 3 in the ABR of people who say they have problems hearing the noise is substantially lower, more than half, compared to people who don't say they have problems. So that's one line of evidence how we think, this is not just some age-related phenomenon where everybody has changes, but specifically those people who experience the central hearing loss have changes in ABR waveforms. And then, importantly, we can do the same experiment in animals. We can take a gerbil, we work with gerbils, we can take a 2-month-old gerbil, we can take a 2.5-year-old gerbil, which are roughly the equivalents to the 21-year-old, 68-year-old human, and we can do the same test. And so that was in collaboration with Dan, and we found something similar. Wave 3, see the difference here. So that's an animal we think experiences hearing loss. The advantage of the gerbil is we can now slice the animal, make brain slices like I showed you earlier, and study the mechanisms, which we can't do with humans because nobody likes to have holes drilled in their skulls or their brain sliced. So we have to use animals for that. And so the ABR is basically the link between the human and the animal studies. What we can also do in the animals is do sound localization experiments with them. So the animal was in a chamber, and there's a loudspeaker on top of the animal, and whenever there is a loud explosive sound, the animal startles. You will too, with a boo. And then there is an accelerometer. Whenever the animal startles, it will measure that startle and quantify it. And so that's not surprising. Also, what is not surprising is if you have a warning that the startle is about to happen, you startle less. I say, now, boo, then you will startle less. Okay, so far pretty easy. And then what we asked the animals, no, we didn't ask them to do anything. What we did is we swapped speakers. We asked the question, can they detect? So there's a series of tones coming out of one speaker, and then just before the startle, it's coming from the other speaker. If the animal can detect the speaker swap because it's warning that the startle is happening, it will startle less. If the animal didn't hear the difference between the two, it will be surprised and will startle. And so that was done with young and old gerbils here. And so PPI is kind of, let's not worry about the math, but the point is the lower these numbers are, the less often animals detected that speaker swap. And so for young gerbils, 7.5 degrees, 15 degrees, 30 degrees, they detected it reliably. They did exactly the same as for control. Older gerbils had problems with that. So if the speakers needed to be 40, 45 degrees apart for them to be able to detect it reliably, and then the speaker swap was closer together, they did not detect it as often. So what this means, if you put this animal in a cocktail party situation in a Friday night bar, that would probably not be able to discriminate between two different sounds that are 30 degrees apart from each other. 30 degrees is pretty wide in a bar that is like 10 other speakers or something. So that means we can recapitulate the same central hearing loss in an animal model. Then we make brain slices. And what we think is happening, there are two changes that are happening. This afferent fiber here, the myelination is decreasing with age, meaning that information is less temporally precise in there. And then these other two pink arrows are inhibitory outputs from this nucleus onto the sound localization nuclei that are also getting temporally less precise with age. And these are the two mechanisms. So if that held up, these are like the main hypotheses that we work right now. If that held up, then we would know exactly what to try to change in an old gerbil to make it hear like a young gerbil in terms of cocktail party effects. And so,
I have a question.
Yeah.
Sorry.
Yeah.
So, like, my dog can hear really well. So does that mean that he or she's picking up really small waves that have gone farther?
So can hear well, you mean you have the dog whistle and you don't hear a thing, and the dog can hear it?
Well, like if there's someone walking down the street with their dog, my dog knows it way before I can hear anything, that dog's coming down the street. So how come she can hear so well?
Is it just hearing, or is it other
Sounds.
things? Is it other things?
Maybe
So
sounds.
so dogs don't necessarily hear better than humans.
Oh,
They hear different frequencies. So if you could hear 10 times better than you can, and 10 times is not a lot because everything is on a log scale, you could hear the air molecules in this room bump into each other. A normal hearing human that has normal hearing, the hearing is right at the physical limit. So I would argue that it would be really annoying to hear the air molecules bump into each other. So it would make no sense for you to hear any better than you can. But you still, the dog whistle, you go like this, and you didn't hear a thing, but the dog responded and obviously heard it. That's frequency.
okay, so it's a
So that's
frequency.
ultrasound. It could be
Okay.
frequency, and bats echolocate. All you see is the mouth open and go like this. You don't hear a thing, but that's the frequency range is outside your hearing. But in terms of amplitudes, I would not say that dogs hear better than humans.
Okay, it's just a frequency.
Yeah,
thing.
yeah.
Interesting.
Okay, so age-related changes, methods of manipulation. Let's talk about a few of these at the end. So if we knew what the changes were in this sound localization pathway, how could we manipulate it? And so one way that we're exploring is optogenetics. And so optogenetics is a technique where somebody stole the DNA for a light-sensitive ion channel from an algae or a bacterium. So these guys live in ponds. And during the day, they come up to do photosynthesis. During the night, they sink down so they don't get eaten. And somebody took the DNA for that ion channel and put it in a virus. And you can buy that virus. And so why a virus? Because a virus naturally infects host cells. If you catch a cold, there is a virus that forces your body cells to express the DNA of the virus and make proteins for new viruses. And this is how you get sick. So if you can re-engineer this virus, and we didn't use, we buy those, to take out all the genes for new viruses and you put in your light-sensitive protein DNA, then you made yourself a virus that can bring light-sensitive DNA into neurons. Neurons are not light-sensitive ever because it's dark in the brain. There would be no reason to be light-sensitive, but we can make them light-sensitive. So we buy this virus and we drill holes in animal skulls and we insert a little glass pipette into the brain area we want the virus to go, and we pressure check virus. And then we wait 2 weeks for the virus to do its thing. And then when you just look at the brain, cells look like this. So the virus, the light-sensitive ion channels are not only light-sensitive to control activity, they're also fluorescent. And so you see there's a lot of these yellow blobs here, and that tells us the light-sensitive ion channel is very well expressed all over this neuron. Then what we can do with this thing is we can turn neural activity on or off with light. And so you could do this in the brain slice. It fires action potentials, then you turn on the light and it stops firing. So that's in the section. And how about in the live brain? You use something like this, and that's what the one core is building initially. Jen started this, and Jamie a few years ago, and there was a CAP center in downtown that became the one core here. So they do things like this. This is an optical fiber that's attached to a laser back here. And this is an implant. So this is kind of like a plug. It unplugs right here. So you can take that out. This is a disposable implant that you can put into the animal and park it right in that area where your light-sensitive ion channels are. And then this sticks out the top. And then when you want to work with the animal, you plug that thing in. And then you can get, in this case, green light down into that brain area. And green light is useful to turn off brain activity. And so you could use this to turn on brain activity, or with a different protein, here, this one to turn off brain activity, and there's a different one which you can use to turn on brain activity. Or you could do both at the same time. You can use 2 viruses and infect the same cell twice, and they would have both types of light-sensitive ion channels in it, and then you shine light of one color, you turn it off, you shine light of the other color, you can turn it on. And so that's a huge field that was invented maybe 10 years ago, and the field has exploded since then. Like, everybody does optogenetics. And it's not only a great research tool, it's also explored for a whole lot of medical conditions. And so, sometime in the future, a lot of people will walk around with glass fibers in their brain where they have electrodes today. And if you do that, so that's where our tech transfer work comes in with Tim Lay. If you do this, the obvious question is, how high do you have to turn up your light so it can do its thing? Or in other words, when you turn on the laser, how far will the light spread and for how many micrometers away from the laser too? Will it affect neurons? And so the people who invented optogenetics at Stanford and MIT, they said brain is brain. And they had scattering coefficients, and they said, here's an equation where you can calculate that. It tells you to turn on the laser so much, and then it will do that. But when we do our brain slice experiments, our sections look like this. So all I want you to see, it's very bright up here, and it's very dark down here. This is the auditory brainstem where we work. And so it didn't look to us as if it is the same. And so we quantitatively measured it, and we found huge differences between these areas. And since the scattering coefficients are in the exponent of an equation, it means that small changes in scattering coefficients have very big consequences on the demands for light. And the differences are so big, if you want to illuminate optogenetic proteins for 1 millimeter away from the fiber tip up here, you can buy a $1.50 LED at some electronic shop, and it'll be fine. Down here, you need a laser that is so strong that it doesn't even exist. Like the strongest laser you can buy for multi-hundred thousands of dollars. It's still not good enough, so it has huge consequences. And based on that, together with Tim, we made this app, which we call the optogenetic app. So it has a whole mouse brain atlas in there. You can download this thing, and then you can put in your parameters for the experiment. So what type of glass fiber do you have? What type of laser? Which optogenetic protein? And then there's a brain atlas where you pick your area, and then it models light scattering for that particular experiment. So that's one thing which, because of which I have to do all the conflict of interest and everything. So it's a pretend company, as
How are
Anna
your
calls
sales?
it. Zero, because the app is free. The app is free, but it is
You haven't downloaded it yet?
it's
I
an
don't
it's
have
an
it on my phone.
it's an LLC. It's an LLC, the company. So it's by definition a for-profit company, although it's a pretend company. And so, but we haven't made anything because we give it away for free. And then another thing is, how about controlling brain activity? So right now we have these big glass fibers that go somewhere. That means the laser is somewhere and the amplifier is somewhere. You have an animal that is tethered. That's really pesky. If you could untether the animal, that would be really helpful. And eventually, if you want to use this in humans, people don't want to walk around with big lasers. You need to make very small devices that don't look bigger than a cochlear implant or something, like a teeny box that you mount to the head. And so that's what we're doing with Chen and Tim and the people in Macau. And so the relevance of Macau here is that they can make printed circuit boards as small as almost nobody else in the world. So in order to make devices small, you need to make circuit boards smaller and smaller. And 20 years ago, that technology was cutting edge here in the US, but it has moved to Asia by now. And so they are at the very forefront of having like 2 wires that are, I think at this point, 7 or 8 nanometers apart from each other. And so that's basically the key to getting cell phones and getting miniaturized devices. And that's the key for us. And that's why we work with them. And so Chen is a student over there, not anymore, he just graduated. But so he makes his designs and then he sends them to Macau, and they have these circuit boards printed every few weeks or something. And then we work with them. Another thing is a chamber that was also done together with Tim. So the brain slices, the brain sections have to be bathed in artificial brain juice, as we call it, to keep them alive. And if you want to buy expensive, if you have to buy expensive pharmacological agents, everything goes by concentration. So if you buy something where a milligram costs $500, if you do that, then you want to be as conservative with using that solution as possible. And if you don't have expensive things, you just use solution and then you throw it away. So you go through a gallon of salt water per day. No big deal because it doesn't cost much. But if you have expensive agents, you can't afford that. And so there is this push to use less and less and less volume for your experiment. And so that was kind of a key innovation where rather than having a circuit where there's a container here and the tube goes in and the tube out, we recirculate, we just fill this chamber here that's between 1.5 and 2.5 milliliters. That's as small as we can get it. There's dishes baked here. And then there's a water pump mechanism that oxygenates the solution and circulates it in a particular way. So that's a patent that was granted last year. And the university owns this, I guess. And I think we're pretty much So to summarize this, so I told you something about, how we study the basic function of the sound localization pathway, how we're interested in how this changes in medical conditions. We focus on age-related changes. Autism is really very similar, like in autistic patients, since you ask to have that same change in the sound localization pathway and the same increase in spatial angle. And that's why autistic patients often get confused when there's multiple sounds active in the same room. And that's why they avoid social situations like that as best they can, because they can't function in that for the same reason. And then we talked about some methods of manipulation, and then I told you about my secret plan to bring all these together. So it would be, if we knew which exact areas, which exact ion channels change with central hearing loss, and we know we're learning how to infect our brain areas with virus effectively, this time for optogenetic, it would be really a small deal to make a new virus that has that protein which gets downregulated with age overexpressed in the virus, and then we try to bring it back, and we would basically try to restore the hearing abilities of a young animal in an old animal. If that works, then we could think about how to bring that into the human. And then, of course, it wasn't me, but there were a lot of people. So here's the lab, and then we worked with Dan, mostly on the ADR testing. Christine Ula and Melinda Anderson at the clinic worked with the human patients, and then Tim Lay, and through him, the University of Macau, we're working on all the tech transfer issues. That's it.
Thank you.
Wow.
Any questions for Achim? Well, thank you,
All right.
Achim.
Thank you,
That was
Achim.
Very
Thank you for coming.
interesting.